So complement system refers to a set of 30 different proteins, and we can include the complement system under the internal defense, and to be very precise, we can include them under the second line of defense. Okay, so along with cellular barriers and cytokine barriers, and we also have the complement system, which forms an important component of the second line of defense. So we had learned that the second line of defense includes cellular barriers and cytokine barriers. You can include the complement system also, which is a set of 30 different proteins which belong to the second line of defense. So please remember, these are proteins, and let us see what exactly these proteins do in our immune system. These proteins were first discovered by Jules Bordet. However, the name was given by a scientist called Ehrlich. Their job is basically the most important job of complement system is to literally punch holes in the membrane of the foreign cells. Say a foreign cell or a microorganism or a pathogen has attacked your body, then these complements are actually activated by antigen antibody reaction. Antibodies are a part of your humoral immunity, not innate immunity. Second line of defense is very much a part of innate immunity. Okay, so whenever there is an antigen antibody reaction, you know that antibody goes and binds to an antigen. So whenever there is an antigen antibody reaction happening, that reaction will trigger these 30, a cascading set of reactions which lead to activation of these proteins, which punch holes in the membrane of the foreign cells, leading to cell lysis. They can also carry out a process where they tag the foreign cell. They go and bind to the foreign cell, and they tell your immune system that, look, here there is a foreign cell. Please come here and carry out phagocytosis. That is also done by their property to activate macrophages and neutrophils. Not only do they go and bind to a foreign cell and show it to the immune system, but they also attract these cells towards that foreign cell, that is by a process called chemotaxis. Sometimes they bring about clustering of antigens together. They bring all the antigens together and create a cluster so that they can easily get degraded by phagocytosis. This mechanism where all of these antigens are brought together and they are forming a clusters, or they are grouped together because of these complements, we call them as agglutination. So the major functions are tagging a foreign cell or labeling a foreign cell, that is called opsonization, and bringing forth the macrophages and monocytes and neutrophils, that is called chemotaxis, and punching a hole by forming a complex in the cell membrane of the foreign cell. It is called membrane attack complex. They form a special complex in the membrane of the foreign cell, thus punching a hole in it. The cell will leak and the cell ruptures and it undergoes lysis, that is called cell lysis, and then gathering together, grouping together all the antigens, which is referred to as agglutination. Again, by grouping the antigens together, you are making it available to the phagocytes so that they can be devoured and destroyed by the phagocytes.